Answers to the first page now on the video. Answers to the second page. Comments, questions, concerns on the first three. I made a mistake, so screw that guy out. Interesting. Okay. Oh. Are you guys looking for my grade 9s that I don't have anymore? Are you guys link leaders? Are you no, you're grads. Who are you looking for? If she's not in there, she's teaching their class. So yeah, feel free. Okay, going once, twice, thrice, no comments, questions, concerns, sold. Here's the bottom one. Okay, hearing no concerns. Next page. Okay, just because you may have something different than what I got doesn't mean necessarily that you're wrong. Let's at least check. Take a look at what you got, compare it with what I got. If there's any differences, throw your hand in the air and we'll talk about it. Probability F1, F2, the phenotypic and genotypic ratios, which we did talk about yesterday. Homozygous, homozygous dominant, homozygous recessive, heterozygous. And to determine probabilities of children, genotypes and phenotypes, and figure out the parents' genotypes. Comments, questions, concerns on any of this? If you didn't do your homework and are just copying things down, shame on you. Yes, Luca.
What's an F1?
That is how we notate what generation we're in. We did that on the very, very first page, like of genetics, way back over here. Where is it? Where is it? When we started heredity, right there. Okay. Next page. There's a teeny tiny Punnett square in the middle there. It asks you to label the different parts and then answer some questions down below about the Punnett square itself. Comments, questions, concerns on any of those? Okay, here's the bottom section. Comments, questions, concerns. See, if there are no questions, either you are all incredible or none of you did your homework. Or it's a blend of incredible and didn't do homework. Yes. Question number 4. You're confused with number 4. So all of the stuff that we've been looking at, when I first introduced the concept of Punnett squares, we specified what type of cross is shown. So if I go back to here, I first introduced the, where are they? In the Punnett squares. Here. Punnett squares with monohybrid crosses. So these are all, and I will show you today what non-monohybrid crosses look like. But that is what that is. What type of cross? It's a monohybrid cross. Mono meaning one. So it's a cross of we're crossing singular traits. One trait with one trait. Okay. Malik, does that make sense? Okay. Next page would be this. Comments, questions, concerns. Sorry? I have no idea. What's happening on Friday? There is an optional assembly during tutorial. I have to pull up the email now. This week, all this week has been the the red dress for the missing and murdered Indigenous women. I don't. Usually they hang something in the rotunda. I don't know if they have this year. I don't think they hung anything. Usually they hang out little like cut out red dresses. I know like City Hall has red dresses hanging in the trees outside. The assembly tomorrow is for that. So I guess you would need permission from whoever's running your B block tutorial because tomorrow's bad C. I don't know if teachers have to take kids down. Any of you who haven't written me the quiz from DNA structure, function, and protein synthesis, replication and protein synthesis, need to be here to write that thing. If you want to go to the assembly afterwards, you're free to do that, but you have to finish. I need you to write me that quiz. So whoever your block B teacher is, if you can come here to instead, that'd be great. Tomorrow goes bad C, so B block is what bleeds into tutorial. Okay, here are the bottom couple questions. Sorry? Okay. Comments, questions, concerns on any of this? Once, twice, thrice sold. Put your phones away, Imparis and Elijah. Maybe not Imparis. Maybe Imparis was just looking like he was on his phone. Elijah was definitely on his phone because I saw it. Here's this page, page number 20. Comments, questions, concerns on any of these? Too many of you are just copying down answers. You need to actually go ahead and do the work yourself. Otherwise, it's not going to make any sense to you. We need to learn the stuff. The videos will be posted. You can always go back and look at what the answers should be. If there are no questions, let's talk about trolls. Here we go. These are my answers to the trolls. Is there anything in there anyone would like to talk about? Anyone who did this or attempted it? Any confusion about how it was done? I'm happy to go through it. Based on the information up top, this is what we are able to deduce. Anyone who would like to talk about it? Once. Sorry? Sorry? Pair one. Yes. Let's talk about pair one. So let me grab my blank one. There we go. So, based on, you want pair one? Let's look at pair one. Pair one has hairy toes and hairy toes. 75% hairy, 25% hairless toed offspring. Kiwana, are you cool with the idea that the dominant gene is the hairy toes? Is that what you said too? Okay, cool. So to determine what each are, we need to look at how we get a 75-25 split. We've done enough of these Punnett squares so that hopefully you guys can see that for 75%, that means we need 3 of the 4 having hairy toes and 1 of the 4 being hairless. If we said that hairy toe is dominant, that means we're going to need a lowercase h down here. And if we have 75% hairy toes, that means we have to have at least one capital H in all of these other three squares. The only way we can have a lowercase down here is if we have a lowercase and a lowercase there. But we have to have these uppercases here, here, and here. So the only way that's going to happen is if both parents, and for both parents to also be hairy, we need them to both be heterozygous. We have to have a lowercase, but three of them have to have an uppercase. The only way that happens is if both parents are heterozygous. So that's going to give you big H, big H, big H, little h, big H, little h. All three of these, 75%, are going to have hairy-toed offspring. And that's going to be your 25% hairless-toed offspring. That only happens when they're two heterozygous. Does that make sense, Kiwa? Okay. If there are no more questions, I would like you to put down your writing implements. And I am going to show you where this actually goes. So, we are not going, what I'm about to show you, like we've been doing Punnett squares for monohybrid crosses. I said at the beginning of all of this that I am going to grossly oversimplify genetics for you. It gets more complicated than this. Where does it actually go from here? Now, what I want to do today is I'm not going to expect you to know and understand what we're about to do, what I'm about to show you, but I want you to see it. Okay. This is the next step in genetics. We've done monohybrid crosses. What if instead of monohybrid crosses, we would like to do dihybrid crosses? Dihybrid just means now we're crossing multiple traits. So instead of having just flowers where one is purple and one is white, and I breed these things together and I get a purple or white offspring, what if I start taking some slightly more complex organisms? And again, you don't put this down, you don't know this, but I do want to show you that it gets more complicated than what I've done. So now we're going to get cats. This teeny tiny little cat up here has the stubby tail, and we've already talked about how the bobtail, what that's called, is a dominant trait in cats. So this guy has the bobtail. If I zoom in here, can I zoom in here? We can see that, let me scroll. We can see that here. This guy has bobtail, and he's white. White's a recessive trait, brown's the dominant trait. The short tail is the recessive trait. The brownness is the dominant trait. So for these cats, this is the female, this is the male, because those are eggs and that's sperm. If this is the genotype, now we can start to split up multiple genes. There's an S gene, a capital S gene, and a lowercase b gene. A capital S gene and a lowercase b gene. A lowercase s, capital B, lowercase s, capital B. And these are nice and easy because each one of these guys are, all the traits here are homozygous. But now, when we're mixing these together, we're breeding cats together with multiple traits. Now, you can actually have multiple combinations of the kinds of sperm that result here. What if instead it's not homozygous, like these two traits aren't homozygous, these two traits homozygous? What if it's not that way? What if we get heterozygous instead? What if we can say, okay, what about brown could also be big B, little b? So what if I have big B, little b, and big B, little b? What if the what if the other trait, the bobtail or not, is heterozygous? Then all of a sudden, if we've got multiple traits, it's not going to be 2 by 2 anymore. Now we're going to be 4. And so we're going to get 16 possible offspring combinations. That looks something like this. And so this is a kind of a crash course on where this comes from. If you recall, back to meiosis. When meiosis happens, the homologous chromosomes line up. And if you're breeding genes that happen to be on different chromosomes, you can have situations where when the homologous chromosomes line up, they could line up. So in this case, we've got parents that are heterozygous for two different traits. I've got an R gene and a Y gene. You can have them line up like this, where the big R and the little y to go together, or the big R and the big Y could go together. And you can now start, because the gametes only have half the possible genetic code, you can start getting combinations that look like this. You can have this as a gamete possibility, this is a gamete possibility, this could be a gamete possibility, this could be a gamete possibility. And so now we're going to start to breed all of these different gametes together. And we start to get some of these ridiculous sized Punnett squares. Stop me at any point if you have questions you'd like to ask about what you're seeing. We have so many. Pick one.
How do you get too little? So you have too little S's, too big P's. How do you get big S?
Like up here?
Yeah.
So this is like the time. So I pulled this. I went to Mr. Easton and I was like, I want to show my students where this goes next. And he handed me this. This is what he used. This is what the pre-diploma 10s do. You guys are not a pre-dip 10. We don't do this in regular 10. The pre-dips do all sorts of ridiculous stuff. This being one of them. So this is, again, being just the title page, this is just an example of what could theoretically happen. So you could have the big S combo, or you could have a combo like this. So the only way a combo like this shows up is if each parent is heterozygous, like it was on the next page. So for this to happen, the female cat would have to be big S, little s, and big B, little b here. And same thing with the male up here. So like down here, that's where, if both parents are heterozygous, so now instead of using S's and B's, we're using R's and Y's. But this code is what it would have to be for this kind of Punnett square to show up. And the way meiosis occurs, they could split up like this, and so you can get all these different combinations from two heterozygous parents. Does that make sense? Does that answer your question, Simon? Now, meiosis happens, homologous chromosomes line up. Let me see you look like you're in pain. Okay. And they get split. One of the other interesting things, I'll forget that for a second. So now we're going to set up some dihybrid crosses. Let's say we did two heterozygote P planes, where we've got R, like heterozygous, R being round, little r being wrinkled, big Y being yellow, little y being green, yellow and round being the dominant traits here. If we took two heterozygote parents and bred them together, this would be the resulting population distribution for the offspring, where you'd get 4, 5, 6, 7, 8, 9. 9 out of the 16. 9 16ths, that's 9 16ths, that's not a reducible fraction, would be yellow and red. Sorry, not yellow and red, yellow and round. 3 of them would be green and round. 3 of them would be yellow and wrinkled, and then 1 of those 16 offspring would be wrinkled and green. And so now when you're starting to breed multiple traits together, you're starting to get a wider distribution of possible offspring phenotype trait, or phenotype. Good. Sort of. Now, yeah, we've done that. That's what we just talked about. So Mendel did this work as well. Rather than just taking monocrosses, he also did the dicross, and he realized that what he was seeing didn't always match with this kind of predictability what he was expecting. Now, so cool. So there is an effect. Again, it depends on who you have for Science 9. I don't think pre-diploma Science 9 did not exist last year, did it? It did? It did, but it does not exist this year. Did any of you take pre-diploma Science 9? Hands up. You're the only one. You did too, with Easton or Morrison? Easton? Easton? Okay. So with Easton, in pre-diploma 9, did you guys discuss recombination as a part of meiosis? Do you remember? You don't remember? you were also created at Einstein? You did not discuss it? Okay. So, in meiosis, when you have, I'll zoom in here and explain something here for you. In meiosis happens when you go, so your homologous chromosome pairs line up, and then each chromosome in the homologous pair gets copied. So afterwards, you get, so in this case, we've identified them by color. We have a white one and a blue one. They both have the A, B, and C gene on them. That's a homologous pair. The white one gets copied. The blue one gets copied. So now we have a homologous pair and its duplicate. One of the fun things that can happen, how this happens, I do not know. I am not a geneticist. One of the fun things that can happen is because of the proximity of these homologous pairs. Okay. One of the fun things that can happen with these homologous pairs, because of their proximity to each other, they can get tangled up. So you can see here we've got 2 white ones, 2 blue ones. The middle pair, because of their proximity, there's an overlap. There's some wrapping that happens because of the physical proximity these guys have within the cell to each other, where they can get close together. The DNA chain in Paris and Vikram can break. And when it fixes itself, they actually latch onto the other one in the pair. So now instead of having 2 whites and 2 blues, you've got 1 white, 1 blue, and then like 2 halvesies. One with the top half blue, bottom half white, and one with the top half white, bottom half blue. And if the genes that you're comparing are far enough apart on the chromosome, when this splitting happens, you can actually start to get this blending, where if you see here, the A's are close enough together so that they don't get split. The B's are close enough, the A and B's are close enough together on this one so they don't get split up. But the C's are way far apart, and so instead of having A, B, little a, little b, little c, you can actually have little a, little b, big C, where that was not a combination that existed on either of the homologous pairs in Paris and Vikram subtopy. At least pretend like you're paying attention. So this is what's called crossover and recombination. If the genes are far apart, or the specific genes are far apart, you can have this happen. If they're close together, it happens less likely, but it can also still happen. And so when this happens, you can start to get crosses that you did not expect coming out of these Punnett squares. And that happens, one of the ways that you guys look at this is if you decide to go off into biology and university and you want to start doing things like this, it takes a long time to breed cats. And people start to get a little bit uppity. Like, did any of you watch Price is Right as a child? Or you stay home sick, you watch Price is Right? None of you? Just me and Lexi, apparently? What was said at the end of every single Price is Right episode? Did you watch it with Bob Barker or Drew Carey? Was it a tall old guy or was it a short fat guy? Who lost a ton of weight? You don't remember, okay, cool. So Bob Barker, old guy, used to run Price is Right. At the end of every single episode of Price is Right, he would say, don't forget, ladies and gentlemen, from our sponsor, the American SPCA, don't forget to spay and neuter your pets. You don't want to just at the end of every Price is Right episode, it was a plug he had for the SPCA. Because you don't want large populations of mammals flooding an ecosystem. So when you're in biology university, you're not going to breed a bunch of cats together. The last thing you're going to do is create massive populations of cats, because you have to get rid of them afterwards, and people frown on putting cats in bags and throwing them in the river, because that's effectively what you have to do. You have to euthanize massive population of cats or dogs. Okay, that's what you're going to breed together. But nobody cares about flies. Flies also breed really, really quickly. So one of the populations that you look at when you do these genetic experiments in universities is you breed fruit flies together. Everyone hates fruit flies, and fruit flies have a short lifespan and they breed like crazy. So you breed fruit flies together. And then they die naturally, and you go through their corpses and you see the kinds of options, and you're like tweezers, like literally tweezers, and you're separating out the different kinds of flies that exist. with the phenotypes that you can see. And so when they're crossing flies, excuse me, when they're crossing flies, you can see that they're breeding different colors here. Gray with normal wings and black with what are called vestigial wings. So the wings aren't formed the same way as they do with the normal shaped wings. And so you can see here, you start to get the breeding here happening. And you start to see combinations, but there are combinations that might pop out that you don't expect. And with large numbers of populations, like I said, fruit flies breed fast with a short lifespan. So you get, the ethics of killing them, you don't have to worry about it. They die around in a couple days. You start to get these effects that start to pop out. There is also some, oh yes. There is also something that pops out in the natural world when you start to look at populations of organisms. This, oh, where's my mouse? There it is. Oh.
Mr. Rios, how long does it take to breed a fly?
I mean, you don't breed flies the same way you breed other animals. If you want to breed dogs, like, there's a certain level of human involvement to breed two dogs together. Flies, you just put them in a jar and they'll figure it out. Are you serious? What are you doing? Are you actually on the phone right now? With who? Then hang out. Why don't pick up a scam call?
Why not?
Because I'm trying to teach you things. Put your phones away. If it's your mom or your dad or someone you expect the phone to be in an emergency, let's talk about it. Scam, put it away. Okay, so one of the other weird things that can happen is you get this sort of scenario. Now, we expect dark fur to be a dominant trait. White fur is a recessive trait. So when you breed in rabbits, for example, another population that breeds very, very quickly, you get rabbits. Black is the dominant trait here, but look at where they live. What's the background color in all those pictures? White. If you're a black rabbit that lives in the Arctic, for 3/4 of the year, there's snow on the ground. What are the odds that you survive that experience? Yeah, basically zero. Not zero. If you're like Superman rabbit, you might get away. But if you're average Joe rabbit and you're black on a white background and literally everything wants to eat you, you're probably going to die. Probably before you're smart enough to figure out how to breed. But you get eaten. So even though white is the recessive trait, white starts to become the population that starts to dominate, even though it's recessive, because the black's getting consumed out. So outside the lab, there's a whole bunch of other factors that you have to take into consideration when you start to look at some of these processes. What is it called? Yes. This is exactly where we're going. What is it called when nature does the selecting? Natural selection. Nature does the selecting for you. So even though you have traits that are genetically recessive, over time, nature can take over and be like, yes, this is the recessive trait, but it's actually advantageous in this area. And so that's the trait that's going to pop out. And then over time, you start to get these populations that specialize given their environment. We call that macro or microevolution. You start to get different versions of different major species that pop up. Comments, questions, concerns on this? Do we see how I've oversimplified certain things and it can actually get way more complicated? Okay. So, should you desire to go on into biology, that is what's in your future. It's cool. It's fascinating. But it's way more advanced than what we're talking about here. So, now, on to today's stuff. Find me this page if you haven't already. Let us talk about codominance, incomplete dominance, and sex-linked traits. Yes, Michael. No, that was not a hand. Okay, cool. Never mind, I take it back. Here we go. I want you, I want you, I want you, I want you, and I want you, and I want you. Sure. Yes. No. I have 2 out already. When one of them comes back, you may go. Okay, here we go. So, codominance. In directly dominant, Soham and Malik and Angel, and whoever else is talking to you guys. Albino populations? Okay. Well, your DNA is what codes you to you. So, I mean, albinoism is it's a pigmentation thing. You can have mutations where the pigment doesn't show up at all. And what that usually leads to is red eyes and shades of white or cream color everywhere. That's albinoism. In some animals, it's like a cream color. It's not white. It's like off-white. Yeah. Yeah, but like if you have an albino orca, for example, it's not white. It looks more like a cream color. But that is the version of white that shows up in green. Oh, weird. Yeah, Malik.
What about like how Michael Jackson like white?
Sorry? Yeah, because he bleached his skin.
That's a different.
Entirely different scenario. Okay. People don't actually know whether Michael Jackson had a disease or did it to himself. He could have had it. He could have developed it because when you see pictures of younger Michael Jackson, he was black, and he looked black, but he looked lighter than his siblings. So there could have been a disease there. And we don't really see any sort of transitioning. Like we don't see so some people have. There are different diseases where your pigmentation can be different on different sections of you, and so it starts to, like, you can have, for example, a black person who has like patches of they look like totally white, or the other way around where you have a light-skinned person with dark sections of pigmentation. I don't know about Michael Jackson. I know it's not what I want to talk about today, so we can talk about Michael Jackson later. May he rest in peace. So, codominance, incomplete dominance. When you have homozygous traits, what gets expressed is really easy. Either both dominant shows up, both recessive shows up, and you got to get the dominant trait or the recessive trait, and you're done. It's nice and easy. When you have a heterozygous scenario occur, these things, codominance and incomplete dominance, can show up depending on the trait. So in heterozygous scenarios, these two things can show up. So, codominance. Codominance is when both alleles, so both versions of a gene, are fully expressed. What do I mean by that? So, here is a picture of a cow. Here is a color version of this same picture, if I can find it. Yes, Block C was all concerned that the cow had been stretched. Yes, the cow has been stretched. Get it? I just grabbed the picture and I stretched the picture. The cow is just slightly too long. Picture's been distorted. I know, but I didn't because I only had this much space on the page. Okay, so stop it. What are you guys doing? Unrelated. So we've got a cow. Here's the cow. This cow, no, it's not related at all. This cow is two colors fully expressed. It's got some red going on. It's got some white going on. It's not a pink cow where the white and red have blended. It is a full expression of both. This is, no, it's not chocolate milk. This is what is called a roan. When the coloring looks like this, if I have to split up the talkative table, I will. I have lots of places to put people. So the coloring that shows up here is what's called a roan, and what it means is that you've got a full expression of red and a full expression of white, and you get this kind of dopply color here. Roan bulls are heterozygous, where both the base color, the red, and white are fully expressed. One allele is expressed as white hair, the other allele is expressed as the red hair. And the way we write this down is because they are both equally dominant, they're expressed using one capital letter for the gene. So in this case, we're looking at gene color, hair color. So I'm just going to use capital H as the dominant form of the gene. So I've got my capital H there. And we use a superscript letter for the alleles. So red and white are both dominant hair color. So we're going to use H to signify that it is in fact dominant. And we're going to use a superscript. Superscript just means you write it like an exponent. Superscript for R, and another dominant with a superscript for white. This is how we denote that both of those traits have shown up. And both of them are allowed to be fully expressed. You get a roan bull by mating a red cow. So this guy, the red cow would be HR, and a white bull, HW. Presumably it could go the other way too. You could get a white cow and a red bull. I don't know, presumably that's fine. And so you'd get something that looks like this. The Punnett square would be HR, and on the left it would be HW. And so then every square on the Punnett square would be HW, It could be HRHW, doesn't matter. Because they're both dominant traits, we write them capitalized, everything capitalized. Then we wouldn't have a superscript. It would just be little h. If there would be a recessive trait in, so good question. What if there was a recessive trait that got in here? I don't know. If red and white are both dominant, maybe there's a third color that shows up as a recessive. It would just show up as a lowercase h. We wouldn't also have the superscript there. Her hand was up first. You may. And when Vikram gets back, when either Vikram or Namra get back, then Gemini, you can go. Well, two of you put up your hands, one right after the other, about going to the washroom. So in this case, if you took a red cow and a white bull and bred them together, then 100% of the offspring would have what coloring? Which we call, it's not red and white, it would be roan. And then whether it's a cow or a bull is male or female. So an example of this in people, flip the page when you're ready, is a disease called sickle cell anemia. I'm going to explain it right now. This whole page is sickle cell anemia. They're bean-shaped. What would a what? Calicos are interesting because, I mean, calicos are complicated for a few reasons. You can't breed calicos with other calicos. There are no male calico cats. There's no female calico cats. I think there's no. I don't think. Yeah, then there's no male calicos. If it shows up, I don't think it is. I don't think it can breed. I know there's something weird about calicos and the male. Okay, Soham, answer yourself. The coloring there, if all three of those colors are expressed, then those three colors would be codominant with each other. orange, what is it, orange, black, and white?
Yeah.
They'd be codominant. There's no blending happening there. Okay, so an example in people is this thing here called sickle cell anemia. That's this blank up top here. Sickle cell anemia. Now, there are a few ways to become anemic. Anemic means you are not getting, your blood is not oxygenating all of you the way it should. You can be anemic if you have low iron. If you are part of the population that bleeds a lot, every 28 days or so, you can become anemic just because you're constantly bleeding for a week or so. There's all sorts of ways to become anemic. One of the ways to be anemic is a genetic disorder called sickle cell. And so what I've got here are pictures of red blood cells. It's not animation, it's an artist's rendering. Typical red blood cells, Malik, wake up, should look like this. They should be circular, basically like an inner tube. Circular with that kind of lower down area in between. And that is what a normal red blood cell look like. Now, if you have sickle cell, some of the red blood cells, or if you're full-blown sickle cell, all of your red blood cells will have this shape instead. You get this kind of, it looks like a bean or a jalapeno or, a sickle, which is a curved tool used by people to harvest grain. Yeah? Yeah, to get rid of these red blood cells. That's what a blood transfusion does. It takes out your blood and puts in proper blood that is normal red blood cells. And you would have to get transfusions fairly regularly because your body is going to constantly be creating these red blood cells improperly. My gut says no. I know if there's problems with your white blood cells, you can get bone marrow transplant, and that would fix that. But I don't know about red blood cells. Treatment for sickle cell anemia. There are gene therapies. So one way to manage it is, as you say, transfusions. Those transfusions would have to be regular. There are what are called exchange transfusions, where you replace red sickle cells with, you just swap out the blood cells. There are what are called stem cell Malik, what are you doing, man? Malik, you good? Okay, go for a walk. You can get what are called stem cell transplants, where healthy stem cells get transplanted into someone who is producing sickle cell blood cells, and then those healthy stem cells can become the cells that produce healthy red blood cells. So you're not necessarily getting rid of all of the sickle cell producing cells, but you're adding the proper production, so you're not full-blown sickle cell. And then there's like full-on gene therapies. There's something called CRISPR, which if you know, so you as an adult, you'll know if you have sickle cell anemia and you experience symptoms of it, which we're going to talk about down here, you will likely know. But you can be a carrier for the gene and not know, because sickle cell is codominant. You can have the sickle cell gene and the regular gene, and because they're codominant, which we're going to see at the bottom of this page, they both express fully. So you have some red blood cells that are sickle cell and some red blood cells that are normal, so much so that you might not actually have symptoms of sickle cell even though you carry the gene. It's a genetic condition that can be tested for. And so if you found out, for example, that, oh, I'm a carrier, and your partner is someone who actually has sickle cell anemia, you're like, oh, my offspring could have a higher likelihood chance of having this. And so what you could do is, let's say you get pregnant. Let's say if you're a carrier and you get pregnant with someone who's also a carrier, maybe neither of you know, They can do this genetic test. The genetic test comes back like, oh yeah, one of the things I can flag for is sickle cell. When that embryo is still an embryo, you find your pregnancy early on. CRISPR is a gene editing technology that they can do to go in and edit the gene code of your embryo. Or if you go through the process of in vitro fertilization, they take sperm, they take egg, they make the embryo, they can edit the embryo before it gets implanted in you. There are huge ethical considerations about doing this. But for things like sickle cell, it could be good to get rid of a sickle cell trait, for example. But let's say, no, never mind. I'm not going to say what I was going to say. Forget that. So there's all sorts of treatments for this, but it is a genetic disorder. So there are also genetic treatments for this. Okay, so all the things I just said are the things that are going to get written down in some of these blanks. So here we go. Sickle cell, it is caused by a specific form of the gene that directs the formation of a particular protein called hemoglobin. Hemoglobin, not hemoglobin, hemoglobin. It's also the next blank. Hemoglobin is a protein in red blood cells that carries, that should be an S there, not a D, carries oxygen. When you have sickle cells, these sickle cells do not transport oxygen effectively because of their weird shape. They cannot pass through small blood vessels. They get bunged up within the cells, within the vessels. So what that can lead to is it can lead to a blockage. If you have blockage within a blood vessel, depending on where that blockage is, it can lead to just, if it's a blockage like for a larger vessel, like the arteries feeding a limb, for example, that limb can start to die. Or at the very least, tissue damage can result. Tissue damage. If that vessel happens to be in your brain or in your heart, it can lead to what's called a heart attack or a stroke. So there's all sorts of wonderful side effects to sickle cell anemia. All sorts of things can go wrong. The allele for normal hemoglobin, so if you don't have sickle cell anemia, the allele for normal hemoglobin is expressed like this. It's 2 letters, HB, with a capital A. I don't know why they chose the letter A, but they did. Vikram, come back to class. Don't turn around, come on back. Gemini, you can go. And the allele for sickle cell is HB with a capital S. So if you are homozygous, HBS, do you like okay, Malik? Okay. If you are full on HBS, ABS, you have sickle cell in its fullest, most awful form. Individuals who are heterozygous, HBA, HBS, have some normal and some sickle cell. These heterozygous people are said to have the sickle cell trait, but they don't experience any symptoms. What this means is, let's put this down here, they are carriers. But may not have symptoms. And you can be carriers for all sorts of diseases. You can be a carrier for the common cold or common influenza, where you have the virus, your body is just not exhibiting symptoms of it for a bunch of different reasons that can happen. But you can then go, I feel fine. I have no symptoms. I come to class, boom. Now all of a sudden, Angel, Malik, Soham, Kiwena, and Numrit all have the flu, and Lexi's sitting there going, what? Well, it means Lexi's probably the carrier who's asymptomatic. She infected her whole table, and so has it herself, but maybe didn't express any symptoms for it. Some people were like this for COVID too. You had it. You didn't know you had it. and so infected a bunch of people with it. If you're heterozygous, let's say we have two heterozygous parents, both have the trait, but both are healthy. Both of these parents are going to be HB big A, HBS, HB A, HBS. You can start to get different combinations. If you're a carrier but don't actually have the full-blown disease, and marry a carrier who doesn't have the full-blown disease, your kids can have all of these possible combinations. You could have HBA, HBA, where you have a child that is totally normal. The blood cells are being formed as the blood cells should be formed. You have a 50% chance of having kids who are carriers, not sick, but a carrier. Or you can have a child, HBS, HBS, that has full-blown sickle cell anemia and therefore will need treatment their whole life, as long as their life is, for this disorder. Now, if you, Angel asked the question earlier, and I listed off some of these treatments. There are medications that can be used to reduce the pain, because there's pain associated with this. And it can improve blood flow. It widens blood vessels, allows blood to flow through easier. Or blood transfusions, or stem cell transplants, or gene therapies. Now, all of these things are available in the developed world really, really easily. In the undeveloped world, or the less developed world, poorer parts of the world, these medications may not be an option, these blood transfusions may not be a thing, and so kids with sickle cell anemia are more likely to die in poorer parts of the world than in others. Angel, you had your hand up. Okay, so the hand is now down. Yes, do what you gotta do. Okay, what are we talking? Are we talking about something unrelated?
It's somewhat related. This is like, it's all talk to this, but also on biology.
It's from the topic of this.
Okay. Okay, so Angel does not have a question. Because your hand was up a second ago, and then I kept talking, and then it went down. Okay. Yes.
Semi on topic.
Okay.
Does water go to your stomach, like your stomach acid, or does it go somewhere else?
Because the same place all your other food and drink goes.
So stomach.
Yeah.
Does it mix with your stomach acid?
Yeah. Everything you eat does that. Okay. Now we're off topic. So that's sickle cell and complete dominance. Let's bring it back. I got one more topic I want to talk about. You probably aren't going to have homework for me today. We're going to do incomplete dominance today. Let's take a look. So, enough. Stop. You can learn about how water gets digested in Bi 12 when you look at digestion. Or you can Google it later. You learn about digestion, yeah, in Bi 12. Digestion of everything. Google it, or wait 2 years. Focus here, please. Okay, so we have codominance in Paris, where both traits show up fully. Now we've got incomplete dominance. Incomplete is the next one we want to talk about. Incomplete dominance is when neither allele can fully cover up the other. And neither is fully expressed. You get a heterozygote again. It's not HY, HET, heterozygote. Shows a trait that is between the dominant and recessive traits. You use the same method for writing the genotype as in codominance. Cross this out. I'm not going to put anything there. I'll show you what I mean in a sec by going through an example called a 4 o'clock flower. I scribbled it off. I'm not putting anything in that blank. We use the same notation. These are also true breeding flowers. We're going to take a true breeding red flower. So true breeding red means when you, I'll go back to the definition for true breeding for those of you who have forgotten. Where are you, true breeding? Here we go. These are plants that reproduce with predictable results. I think Luca might need to, yes. Luca, if you're squinting in there. I know some of it's messy.
No, what's
Yeah,
what's this? What's after codon?
you can't leave that.
I scribbled out the blank. There's nothing there.
Oh.
I said I'm not writing anything there. That blank shouldn't be there. Scribble it out. Okay, so 4 o'clock flower is true breeding. Means it breeds with predictable results. We're going to take a true breeding red flower and we're going to breed it with a true breeding white flower. Now, the traits here are incompletely dominant. And so we use the same notation for incomplete dominance that we do for codominance. So the red flower is going to be C capital R, C capital R. C for color, R for the dominant trait. Luca, don't write on things that don't belong to you. The white one is going to be the same, except it's going to be CW, CW. Now, instead of dominant recessive, Vikram, pick it up off the ground, whatever you just threw at Memphis, and throw it in the, get up, pick it up, throw it in the garbage. We're going to have CR, CR, and CW, CW. I'd also like you to take your desk and put it back to where it was, because I told you yesterday, if you can't sit there and learn, you can't sit there, so put your desk back. And we're going to get CW, CR, CW, CR, CW, CR, and CW, CR. Now, if these are incompletely dominant, all of these are going to produce a trait between white and red. What happens when you blend, artists, red and white? The blend of red and white equals pink. It's like I picked that color of pen on purpose. Let's make this more interesting, shall we? In the spaces on the left and the right of that one in the middle, I'd like you to draw me 2 more Punnett squares. Boom, boom. Let's breed some more colors, shall we? What if I told you that you had a heterozygous green flower? To be a heterozygote who's green means what had to be blended together to get the green? Blue and yellow. So if you're green, that means you had to have blended a blue with a yellow to get that color. Now, if you're going to take a green one and we're going to breed it with a pink one, pink had to be CWCR because, again, the incomplete dominance means that the colors are going to blend. I don't know. It's the short answer to that question. So, let's breed these things together. We're going to get all sorts of crosses. CB, CW, or sorry, CW, CB, CW, CY. What are these going to result? What happens if you take blue and you add some white to it? Light blue. What happens if you take yellow and you add some white to it? Yeah, you're going to make light yellow. It's just going to be faded or muted. But what happens if you take the red and you get it mixed with the blue? You're going to get purple. And yeah, the last one, CRCY, is you're going to get some offspring that are orange. And now all of a sudden, we get some cool things resulting. What happens if we take, let's go back and do the simpler one. I bred the red and the white, I got a bunch of pink. What if I take the pinks and I breed them together? So I'm gonna get CW, CR, CW, and CR, and we're gonna breed those together. Well, this top left box is gonna be what? Which is gonna be white. That white color comes out. What if I breed, so this box is gonna be CRCW, this bottom one is gonna be also CRCW, so the diagonal then is gonna be more pink, and that bottom one is gonna be CRCR, and so that one's gonna be red. There's no reason you couldn't. If those combinations exist, yeah, there's no reason why you couldn't. So you wanted CB, CY with what? Oh, that's what we did over here. And then we got the purple, orange, light yellow, light blue. The interesting here thing that though results is, if you wanted to figure out what those dominant base colors are, you would just have to take whatever color you got and breed it with itself. Because if you take, it's like you've got these pink flowers. If you breed pink flowers with themselves and you got half of them are pink, but a quarter of them are two different colors, that means to get the pink, you started with these the two outlier colors. If you wanted to figure out if green was a blend, you took a bunch of green and bred them together, you'd get some that were blue, some that were yellow, but 50% would maintain their greenness. What that would mean is the base colors there would be yellow and blue. Those would be the fundamental base colors. And that's how you would determine what the genotype, Malik, I don't know what you're doing, but please stop it, would show up to be. We've got a little bit of time, so let's start this piece. Let's talk about sex-linked traits. We've got incomplete, we've got codominant, and now we're going to lump in here the concept of sex-linked traits. We'll see how far we can get. Traits that are controlled by genes on the sex chromosomes. For those of you who aren't sure what those are the X and Y chromosomes. Traits for genes on these chromosomes are called sex-linked traits, because whether or not they are expressed is connected to the fact about whether or not you are biologically male or biologically female, or genetically male or genetically female. And we talked, I can't remember if I talked about, we talk about color blindness with you guys, or was that the other block? Okay. Genetic males are affected by recessive X-linked traits more often than females. Anyone want to take a guess as to why that is? Yeah, because they have one X. Males have one X chromosome and one Y chromosome. And so if a recessive trait shows up on that X chromosome, there's nothing stopping it from appearing as a phenotype. But in women, women are less likely to see the recessive because you, genetic women, have two X chromosomes. So in order to see a recessive trait show up in you, both X chromosomes have to have it. So women are less likely to express recessive X-linked trait because two recessive X chromosomes would be needed. So there's a chance that the dominant allele on the other X chromosome shows up. You ladies have a fail-safe for some of these X-linked recessive traits. Guys, you don't, because biologically speaking, gentlemen, you're less important.
Yeah.
We don't matter, because you don't need a bunch of dudes to pass on genetic information. You only need a couple, but you need a ton of women in order to have All the children are required to pass on the humanity. An example of this is color blindness. We have some time. Flip the page. Now, this color, this picture is photocopied in color. If you cannot see, and it's a watermelon. I'm screwing with you. That's not photocopied in color. This is photocopied in color. Because I don't have, there's no such thing as a color photocopier. I have a color printer. My copies can come out of that machine in color. But your guys are all black and white. So this is what this is supposed to look like. If you are not colorblind, the picture on the left is the picture you will see. That is a watermelon. The inner bit there is pink. The outer bit there is green. If you are colorblind, if you don't see that as being pink, that's pink.
That's red. I'm not even.
Okay, well, whatever. Red, pink, same thing. The point is, Soham. The point is, you see a difference between the inside and the outside.
Yeah.
If you don't see a difference between the inside and the outside, congratulations, you just found out you're colorblind.
I don't see any.
Actually, honestly, no, but like, why would we joke? Don't joke about it. Like, if you actually don't see a difference, and there are people who learn this. Like, I had a buddy who went to electrical school. In electrical school, there are different colors that are present on different devices to denote meaning. This one will have like red stripes and green stripes and blue stripes, and this one will have maybe blue and yellow stripes. So, and there's a whole code system that is used to determine which piece you are actually supposed to use. I got a buddy who did not know he was colorblind until he showed up there and said, guys, these two resistors are the same. And the rest of his class went, nope, those are definitely different. And he said, oh, crap. And that's how he learned he was colorblind. Colors are an interesting one because colors are shaded. Like, you might be colorblind your whole life. You might experience, someone might tell you, oh yeah, that's green, that's red. Okay, cool. You say yes. Because as a child, you are taught based on someone else's perspective of the world. It's like, what is salty? You only learn what's salty. Someone hands you something and say, what you are about to experience is saltiness. Here, lick this or eat this. And you're like, oh, I've experienced this sensation. The sensation must be salty. Or like, I'm teaching my son colors. Jack, this is red, this is green. Okay, cool. If he starts to mix those up, that's indication that he then might be colorblind. Because he's just being taught based on what I perceive. And I'm not red-green colorblind because I see the difference between red and green. Yes, unrelated. Yes, unrelated. Bees see ultraviolet, we don't. Unrelated. Unrelated. It's loosely related. Okay. Why don't you ask, Numrit? Okay. We'll continue with the colorblind example tomorrow. You have no homework from me. You're going to get a buttload of it tomorrow. It's going to be great. Did you say black guys don't exist? Oh, black lights. Like, this is, clearly we have a problem with Malik. If black guys don't exist.